Hi everyone, welcome back. So this is our DNA molecule again. And if you notice these two ends of your DNA molecule, they have some kind of difference. I mean, you can't notice in this diagram, but if we zoom into the structure of our DNA and see how these nucleotides are connected, then there is some kind of difference between ends of your DNA, which gives uh, names to these ends based on what difference is there. Now, to uncover the details about this difference and how we name these ends of the DNA, we need to revisit the structure of our nucleotide. So I told you that we have one sugar ring, one phosphate group, and a base in my nucleotide, right? Now, actually, we have one oxygen atom also, which is present in this five-member sugar ring, and rest of the atoms in the sugar ring are carbon. So this is a carbon, this is carbon, this is carbon, this is carbon, and also we have one more carbon atom between this phosphate group and the sugar ring. And there is a convention which numbers these carbon atoms, which starts from the carbon atom which is connected to this base. So this carbon atom is directly connected to the base. So we'll start our numbering from here and go in the clockwise manner. So this is my carbon number 1, carbon number 2, carbon number 3, carbon number 4, and carbon number 5. Now, keeping this numbering of carbon atoms in mind, if we look at the diagram of DNA, which is something like this. So I want you to visualize these two series of nucleotides again as those two strands of your DNA. So this will be your one strand of your DNA. And similarly, this is your again different strand of your DNA, right? Now I want you to notice what difference we have in the ends of a strand. So let's first focus on to this first strand only. Focus on this particular strand. Forget about this strand. We'll focus right now on the first strand only. So in the first strand, at the leftmost end, we have this nucleotide, right? And in the rightmost end, we have this nucleotide, which is terminating this chain, right? And if you see in the rightmost end, this third carbon atom is engaged with the uh, phosphate group of previous nucleotide. So this third carbon atom is not free; it is making bond with phosphate group of different nucleotide. But in the leftmost end, this third carbon atom is free and it has the capabilities to bond with another nucleotide if we add one more here. But right now it's free. And also, this third carbon atom is pointing outwards in this direction, right? And in this end, third carbon is busy, and fifth carbon atom is pointing outward, right? So you can see that there is a difference between nucleotides of leftmost end and rightmost end. Here in this side, third carbon atom is pointing outside, and at this side, fifth carbon atom is pointing outside. And also, third carbon atom is free in the leftmost end, and third carbon atom is busy in making bond in the rightmost end. So the end in which third carbon atom is free or it is pointing outward, that end is called 3' end. And the end in which fifth carbon atom is pointing outward, that end is called 5' end. Right. Now if I ask you in this second strand, I want you guys to identify in this strand what name should I give to this particular end of my uh, nucleotide series, and what name should I give this end of this strand. You can pause the video and think of what name should I write here and what name should I write here. So if you notice, uh, third carbon atom is busy here making bond with previous nucleotides, but fifth carbon is free and pointing outward, so this is my 5' end. Similarly, in this side, third carbon atom is pointing outward and is free, that's why this is my 3' end. And there is a convention which defines the direction from 5' to 3' end. So if we define our direction from 5' to 3', and then you will draw a direction vector uh, like this, right, for this particular strand. And for the second strand, you will draw a direction vector like this, right? And the one thing that you notice, that two strands of your DNA molecule are anti-parallel. One is going from 5' to 3' in this direction, and the second strand is going from 5' to 3' in this direction. This is always true. In any DNA molecule, the two strands are always anti-parallel to each other. And you know what ends we have in the DNA molecule and how we name them. So that's all for this video.